Hi students, welcome to Bio 11 Lab Week 2. I want to get started with a few administrative things. I just want to make sure that everybody has a copy of both books, the lab manual and the Plato to Darwin to DNA. And if you have those books, if you've created an account for the online quizzes with the Plato to Darwin to DNA book at www.grtep.com. If you need any help with either of these things, just shoot me an email and we'll work on it. So today we're covering two units, the metric system and microscopy. So first, the metric system is a base 10 system of measurement, and it is universally used in scientific laboratories. So many countries across the world use the metric system. The United States, I think, pretty much stands alone in using our standards of measurement, feet, and other things. So we like the metric system because it makes conversions very, very easy between units because it's a base 10, which means that we get to just add zeros or move decimal points. So the base metric unit of length is known as the meter. And so this is sometimes denoted by a small case m. And you can divide the meter into smaller subunits. The centimeter or the millimeter are probably two units of length that you're very familiar with because you see that on a ruler. But then you also see micrometer or nanometer, which are very, very small units of length. And so these become important when we talk about microscopy because we're looking at such small samples. When we're measuring, we are actually measuring in micrometers. So converting units. So if you look at the information on that previous table, you can basically convert the units by either multiplying or dividing by a factor of 10. So if you're moving from centimeters to millimeters, you're going to multiply by 10. If you're moving from meters to centimeters, you will multiply by 100. And if you're moving from kilometers to meters, you will multiply by 1,000. Or you could just move the decimal point. It's basically whatever is easy for you. So I've posted a link to this video on TED-Ed, which shows you basically how to do all these conversions. I found it very useful. This is how I always do my conversions. I always move the decimal point. So check it out. It's very helpful. It'll help you with these conversions. And he's much better at explaining it than I am. Metric units of weight. All right, so the base unit of weight in the metric system is known as the gram. Another important unit that we frequently see in scientific lab and also in life is the milligram. So if you've ever, taken medicine, you would probably notice that your dose is usually in milligrams. So 1 gram is equal to 1,000 milligrams. And then just for a real life reference, so you can understand sort of what a gram represents, a paperclip weighs about 1 gram. The base metric unit of volume is known as the liter. And the most often or most frequently encountered unit is also the milliliter in the lab. So 1 liter is equal to 1,000 milliliters. So when we're measuring liquids, one thing to be very, very careful about is that you must read the meniscus. So if you see here in my image, you'll see that in the cylinder, water will form this curve, this kind of concave shape. And this is because of the hydrostatic forces of the water and the side of the cylinder basically interacting. It's basically static. So when you're measuring, you don't want to measure the side of the cylinder. You want to measure the bottom of the meniscus. So you want to make sure that you get level with that meniscus. If you're actually ever in the lab using a cylinder, you need to get on eye level with the fluid and read it correctly. And this is very important. Otherwise, you can make mistakes. All right, so metric units of temperature. So this is probably where we most frequently see the conflict in the two systems are in Fahrenheit and Celsius. So I have my sister-in-law is from Brazil, and so we are always translating the temperatures to Celsius for her because she grew up with the metric system. and Fahrenheit still doesn't really compute to her. So both of these systems use degrees, but each unit is slightly different, meaning it's just valued a little bit differently. So the conversion equations here for 1 degree Celsius is equal to 1 degree Fahrenheit minus 32 divided by 1.8. 1 degree Fahrenheit is equal to 1.8 degrees Celsius plus 32. I will not ask you to make conversions in temperature. I won't do it. But you know, they both use degrees, so they're called the same thing, but they are different. Something to be aware of, though, is a couple of few important temperatures to remember. So in Fahrenheit, the boiling point of water is 212 degrees Fahrenheit, but in the metric system, it's very easy. It's 100 degrees Celsius. Beautiful. And then the freezing point, we have 32 degrees Fahrenheit, but in the Celsius system, it's 0. So we have a 0 to 100 for the liquid phase of water. So this just makes everything, I think, a lot easier. Some other important metric temperatures to remember is 25, which is generally what we use as room temperature. That's equivalent to about 78 degrees. And also the human body is 37 degrees Celsius. So that's equivalent to about, 98 wherever we are Fahrenheit. Okay, so microscopy. So I had shot a few videos to show you, but the microscope at Queens College right now is actually down. So I'm not going to be able to show you videos on that today, which is pretty sad. But we'll still go through microscopy, and I'll be able to show you some cooler things when we go through mitosis and meiosis lab, because those were also heavy microscope labs. So in microscopy, when we're looking at something under a microscope, the way that you're cutting a sample is going to make a huge difference to what you see. So here I have 3 different ways that you can cut a sample. So we have a cross section. So this is basically a blood vessel, it looks like. We have a cross section, and the way the view of that sample is going to be completely different from what you do in a longitudinal section. And it's also going to be different from an oblique section, which is this diagonally cut section. So when you're preparing a sample and when you're analyzing a sample, it's important to remember how you cut that sample and prepared it so that you will understand exactly what it is that you're looking at. So types of microscopes. All right, so we have 2 microscopes that we usually use in the lab. The first type is the stereo microscope, or also called a binocular dissecting scope. I generally call it a dissecting scope because this is the kind of microscope that I work on in the lab all day. So it generally magnifies at low power, and it'll light specimens from either above or below. If you see here on the stage, there's a light here. So this is very similar to the scope I use in my lab, and it illuminates from the bottom so that you can see through the sample. So this is good for viewing 3-dimensional or opaque objects. So you can take something and place it here and look and get a closer look. You can also see, some, translucent objects, but it really is the best for, 3-dimensional opaque objects that you haven't manipulated or needed to cut. So then there's also a compound light microscope. So this works a little bit differently. It can be either a binocular head like you see here, or it can be monocular, which is as seen below, where there's one eyepiece. Compound light microscopes generally magnify at a higher power than a stereo microscope. Light is transmitted through the specimen from below. So if you see here, there's a mirror here. So this is what reflects the light up and through the lens. So light is transmitted through the specimen from below, and so specimens must be very thin or translucent. So this is good for using cells, thin slices of tissue. In the lab, we usually do a wet mount to look at pond water, and we look at this on our compound light microscope. All right, so parts of a microscope. So here at the top we have, this is the ocular piece, the eye lens. This is where you look in. Then we have the body tube. We also have the revolving nose piece, and on that revolving nose piece we have the objective lenses. So these lenses each have different magnifications. Generally it's about 4x, 10x, and 40x, or 10x, 40x, and 100x. And so you can move up in magnification as you work by switching these objectives. So we have the arm, and we also have the stage, and this is where we place our slides. We have the stage clips. The diaphragm here is how the light passes through, and then we have the light source. And then we also have the base. And then importantly in a microscope, we have the coarse adjustment knob and the fine adjustment knob. So the coarse adjustment knob basically just means that it moves the stage more rapidly. And the fine adjustment knob makes very, very tiny adjustments to the stage. So when you're working in low power, and these low objectives, this lens is not very close to the stage. And so you can use the coarse adjustment knob to adjust your microscope. But when you're working with these higher power objectives, like 100x, that lens is very, very close to the stage. I mean, it is very close to the slide. So you cannot use the coarse adjustment knob. You have to only use the fine adjustment knob, because if you use the coarse adjustment knob, you're going to break your microscope. Basically, the lens is going to crack the slide. So it's very important. Coarse adjustment knob in low power, fine adjustment knob only when you're in high power that you are using. And you multiply these, and that's how you get total magnification. And so here we have basically just a general schematic of the light path going through a microscope to generate an image. All right, a little bit about electron microscopes. So we don't have an electron microscope at Queens College, but these are very, very powerful instruments in research. So electron microscopy is a super resolution method of microscopy. It allows scientists to visualize nanoparticles, which is, you know, super small, 10 to the minus ninth. So rather than using lights, like our compound light microscope, these microscopes use electron guns, which is pretty cool. So there are two different types of electron microscopes, and these have important differences. So we have a scanning electron microscope, which is used to visualize whole specimens. And so I like to compare this to our stereo microscope, because the electrons are aimed at the surface of the specimen and then bounced off, so we can visualize the surface of the specimen. And if you see here at the top, we have pollen grain under scanning electron microscope on the right. So you can really see the texture of the surface of these pollen granules. And then there's also a transmission electron microscope. And this is used to visualize thin specimens, very similar to the way that we use our light compound microscope. And so the electrons are actually passing through the specimen to the camera. So on the right-hand side, we see a pollen grain, which is visualized by a transmission electron microscope. As you see, it's a cross section of this pollen grain. You're not seeing the outside. You're seeing these internal structures. So it has to be very, very thin. And so here below, I just have a little bit of a diagram to kind of give you an idea of what we're actually able to see with electron microscopes and light microscopes. So with the naked eye, we could see about down to 10 to the minus 4 meters. With a general light microscope, we could see about 10 to the minus 7 meters. And then with the electron microscope, we're looking at, a nanometer or more. So these are pretty cool. If you ever, if you're interested, there are a couple different resources on electron microscopes. I might post something, just in case you're feeling like you want to take a deep dive on this. But if not, don't worry about it. It won't be on the exam. All right, so for this week's lab, check out the video on TED-Ed on the metric conversions. I also posted a trailer on Blackboard on Protus because I was really bummed that we can't do the wet mount together. And I went out and I tried. I actually went through a walk in Casino Park and gathered some samples, and I was gonna, look at them for you, but it just didn't work out this week. So I'll hopefully have a nicer video for you next week. So as I kind of mentioned, my PhD is largely based on microscopy. So I'll be giving you a little bit of taste of my research work when we talk about meiosis and mitosis, because I could actually show you some of the images that I've taken. Um, so if you have any questions or you're interested in anything or want more information, please don't hesitate to give me a shout on email, or you can post on the discussion board. And let me know if you have any questions about completing the lab. As I did before, I'm going to post the questions that you need to do in the lab sections, but really just use basic common sense. If it's data that you would have had to gather in the lab, I know that you're not going to be able to collect it, and I'm not going to be grading that. So I really just want you to focus on the conceptual things. Um, learn the parts of the microscope, learn the vocabulary, learn how to do the conversions. Um, and just we'll get through it. All right. So I hope you all have a nice holiday weekend. It's supposed to be some very nice weather, so I hope you can get outside for a little bit. All right. Until next week. Goodbye.